Bismillahirrahmanirrahim. Assalamu alaikum. Today we're going to talk about the DNA synthesis inhibitors, and so in this topic we're going to talk about the antibiotics quinolones. Quinolones are synthetic, broad spectrum bactericidal antibiotics, that is, they have a killing effect over the bacteria. They are effective against both gram positive and gram negative organisms, thus making them broad spectrum. The first quinolone was nalidixic acid, and it was synthesized in the year 1962. Later, a fluorine group was added to the nalidixic acid, and so fluorinated analogues of the nalidixic acid were made, and they were thus called flouroquinolones. These quinolones are used in human and veterinary medicine to treat bacterial infections, as well as used in animal husbandry. Here we have the structure of quinolones. It has a double ring structure composed of a benzene and a pyridine ring that are fused together. The benzene ring contains 6 carbon atoms, while the pyridine ring contains 5 carbon atoms and a nitrogen atom. Here we can see the structure of nalidixic acid. The carbon atom is replaced with the nitrogen atom, and these side chains are added, thus making a structure of nalidixic acid. Then a fluorine group was later added at the 6th position at carbon 6. Here a fluorine group was added, and so the fluoroquinolones were converted to the quinolones were added to converted to fluoroquinolones. The R groups, these R1, R2, R5, R7, R8, they indicate possible positions for structural modifications. Structural modifications can be made at these R groups, at these positions, and so new generations of fluoroquinolones are synthesized. The X usually corresponds to a carbon or a nitrogen atom. Let's talk about the classifications and the spectrum. First generation fluoroquinolones, the quinolones, first generation quinolones include drugs like nalidixic acid, pipimidic acid, and cyanoxacin. These are the oldest and the least often used quinolones. They have a narrow spectrum. They are effective against gram-negative bacteria, mainly against Enterobacteriaceae. Their use has been restricted to the treatment of uncomplicated UTI. The second generation fluoroquinolones include antibiotics like ciprofloxacin, enoxacin, lomfloxacin, norfloxacin, and ofloxacin. They show an increased activity against gram negatives compared to the first generation quinolones. The second generation fluoroquinolones show an increased activity against the gram negatives, including the Pseudomonas aeruginosa, and also against many gram positive bacteria, but excluding Streptococcus pneumoniae. Ciprofloxacin, which belongs to the second generation fluoroquinolone, is the most potent fluoroquinolones against Pseudomonas aeruginosa. The ofloxacin is active against Mycobacterium leprae and Mycobacterium tuberculosis. Other than ofloxacin, drugs like ciprofloxacin, levofloxacin, and sparfloxacin are currently the most commonly used against Mycobacterium tuberculosis, and so fluoroquinolones have come to play a very important role in the treatment of tuberculosis, particularly the multi-drug resistant tuberculosis. So these fluoroquinolones, the second generation fluoroquinolones, are used for the treatment of tuberculosis, and also for the treatment of uncomplicated and complicated UTI and skin and soft tissue infections. The third generation fluoroquinolones include drugs like levofloxacin, gatifloxacin, and sparfloxacin. Although they retain broad gram-negative coverage, all others except the levofloxacin are less active against Pseudomonas aeruginosa than ciprofloxacin. Among the second generation and the third generation fluoroquinolones, ciprofloxacin is found to be the most potent one against Pseudomonas aeruginosa, and except levofloxacin, all others have lesser activity as compared to ciprofloxacin against these Pseudomonas aeruginosa. These third generation flouroquinolones show expanded activity against gram-positive organisms, particularly against Streptomyces and Staph aureus, and also against atypical pathogens like Mycoplasma and Chlamydia. Because of their expanded antimicrobial spectrum, these third generation flouroquinolones are useful in the treatment of community-acquired pneumonia, acute sinusitis, and chronic bronchitis. Fourth generation flouroquinolones include drugs like moxifloxacin, delafloxacin, and trovafloxacin. They show a significant anaerobic activity while maintaining the gram-positive and gram-negative activity of third generation quinolones. Their clinical uses are the same as that of the previous generations, but they also include intra-abdominal infections, nosocomial pneumonia, and pelvic infections. Mode of action. Quinolones enter the bacterium by passive diffusion through porin channels. We know that porins permit the passage of molecules such as nucleotides, disaccharides, peptides, amino acids, vitamin B12, etc. Similarly, drugs can also enter the outer membrane of the gram-negative bacteria through these porin channels, and so these quinolones, they enter the porin channels by passive diffusion. Once inside, they inhibit the replication of the DNA by interfering with the action of two enzymes, Topoisomerase II, which is also known as DNA gyrase, and Topoisomerase IV. So let's first talk about the DNA gyrase. The DNA gyrase is composed of two subunits, gyrase A and gyrase B. It has a ligase domain which is responsible for joining of the two molecules, and also a nuclease domain which is responsible for cleaving the chains. So to understand the concept of DNA gyrase and to understand the mode of action of these flouroquinolones against these drugs, against the DNA gyrase, let's first have a look at DNA. So we know that DNA is made up of a double helix of two complementary strands, just like here, and these complementary strands are supercoiled to pack into a small space. During replication, these strands are separated, like we can see here. When replication occurs, these strands are separated, and each strand then serves as a template, resulting in a semi-conservative replication. Each serves as a template, a new strand is made, and so there is one new and one old strand, and this results in a semi-conservative replication. Unwinding of the DNA results in an overwounded region in front of the progressing replication fork. So for replication to occur, and for these strands to get separated, this area gets overwounded, right, in front of the replication fork. To understand this concept, let's look at this short piece of strand. Suppose we have a rope, a short piece of rope with two strands, just like here, and these are tightly wound around each other. If the strands are then separated, like here, from one end, the rest of the rope will be supercoiled, and a time will come when the overwounded region will make it impossible for the strands to separate. Similarly, in case of replication, the supercoiling in the overwounded region will make it impossible for replication to continue. These strands, because of this overwounded region, these strands will then no longer be able to get separated, and replication, it will be impossible for replication to carry on. So to remove this overwounded region, to remove this supercoiled area, to remove this positive supercoiling, here comes the DNA gyrase. The DNA gyrase then cuts one of the double helices, just like here we can see by the help of the nuclease activity. The DNA gyrase cuts one of the double helices and then passes it through the other, like here, passes one end through the other, and then reseals it with the help of the ligase. It reseals the break. So this action relaxes the positive supercoils and allows the superhelical tension ahead of the polymerase to be released so that the replication can continue. Then we have the second enzyme, that is the topoisomerase IV. The topoisomerase IV is responsible for unlinking DNA following the DNA replication. The double helical nature of the DNA and its semi-conservative mode of replication causes the two newly replicated DNA strands to be interlinked, just like here. When the replication occurs, the newly replicated strands, newly replicated DNA strands are interlinked, and so they need to be separated. Like here, they need to be separated. These links must be removed in order for the chromosomes to segregate into daughter cells so that the cell division can complete. Unless both these new DNA these DNA strands, they get separated, the cell division will not be completed. So then comes the topoisomerase IV. What does topoisomerase IV do? Topoisomerase IV acts like DNA gyrase, and just like gyrase, it is able to pass one double strand of DNA through the other strand of DNA. The topoisomerase IV attaches to one of the strand, and then it cleaves it from here, and then just like gyrase, it allows this DNA strand to pass over the other so that both these strands can get separated, and then this topoisomerase then it detaches, and so two new daughter strands are formed, which are then segregated into new daughter cells, and cell division is finally completed. So what does quinolones do? Quinolones bind to DNA gyrase and inhibit its ligase activity while leaving the nuclease activity intact. The quinolones, in case of the DNA gyrase, the quinolones bind to the DNA gyrase, and so it allows the nuclease. It allows the nuclease to perform its activity. It allows the cleaving to occur, but it inhibits the DNA ligase to perform its activity, and so these cleaved DNA strands cannot join together. And so because of that, because of the this activity of the quinolone, DNA it leads to DNA fragmentation, and so the cell obviously dies. In case of topoisomerase IV, quinolones bind and block their action, thus inhibiting DNA unlinking, and so formation of the daughter cells. The quinolones does not allow binds to the topoisomerase IV, and so this unlinking cannot occur, and so new daughter cells cannot be formed. As a result, the quinolones, they do not allow the DNA gyrase to perform its action and the topoisomerase IV to perform its action, and so replication is not possible and new daughter cells are not formed. Mechanism of resistance. The mechanism of resistance that is acquired by the bacteria against these quinolones. The first mechanism of resistance that the bacteria has acquired is the alteration of the target site. Chromosomal mutations in the genes encoding for the two enzymes, the DNA gyrase and the topoisomerase IV, results in amino acid substitution in the enzymes. The target sites are altered in both the enzymes, the DNA gyrase and the topoisomerase IV. The target sites are altered, and so because of this alteration, the drug becomes inactive. Because of the alteration of the target site, the drug can no longer bind to the target site. It can no longer bind to DNA gyrase or to topoisomerase IV, and so obviously the drug becomes inactive. Second mechanism of resistance that the bacteria has acquired is the porin mutation. Alteration of porin proteins in the outer membrane of gram-negative organism results in decreased permeability of the drug. The porin channels are mutated. There is alteration in the porin proteins, and so the drug that was able to enter by passive diffusion through the porin channels, it can no longer enter easily. The permeability is decreased, and so the drug cannot reach the targeted site, and obviously if it does not reach the targeted site, it will no longer be active. Third mechanism of resistance that the bacteria have acquired is the efflux proteins. These efflux proteins, the mutation enhances the organism's efflux capability, increasing the amount of drug pumped out of the cell. Another mechanism of resistance that the bacteria have acquired is the protection proteins. So QNR proteins are protection proteins that are capable of binding and protecting DNA gyrase and type 4 topoisomerase from quinolone inhibition, and this is a plasmid mediated resistance, and it has become a worrying problem among Enterobacteriaceae and Mycobacteria. These protection proteins, they protect the target site of these quinolones, the DNA gyrase and the topoisomerase 4, and so the quinolone no longer remain active, no longer remain able to bind to the target site and perform its activity. Another mechanism of resistance that the bacteria have acquired is the inactivation of the drug. Inactivation of ciprofloxacin and norfloxacin by N-acetylation of the amino nitrogen of the piperazonyl ring has been observed. This is the piperazonyl ring, and at this amino nitrogen acetylation occurs, amino acetylation occurs of this amino nitrogen, and so this results in inactivating the drug. The drug is inactivated by the acetylation of this amino nitrogen at this ring, of this piperazonyl ring. At this position, the drug becomes inactive, and so it obviously it cannot inhibit the DNA gyrase or the topoisomerase 4 from performing its action, and it no longer remains active. Pharmacokinetics. Quinolones are well absorbed following oral administration with moderate to excellent bioavailability. Bioavailability is a subcategory of absorption, and it is the fraction of an administered drug that reaches the systemic circulation. And so these quinolones, they are well absorbed following the oral administration, and so they can easily reach the systemic circulation to perform its action. However, although they are very well absorbed by following the oral administration, but they interact with cations such as aluminium, calcium, magnesium, iron and zinc, and so if these cations are present in our food, in our diet, and if the drugs are administered along in the presence of these cations, then there will be reduced absorption and the bioavailability will reduce. It should be avoided. These drugs should be avoided to be taken in synergistically along with these cations. These flouroquinolones are widely distributed throughout the body, and they have high tissue penetration, thus being active against intracellular organisms like chlamydia and legionella. Because of its good penetration into bones, orally administered ciprofloxacin is a useful alternative to parenterally administered antibiotics. Parenteral administration is any route other than oral administration. Mostly it is the injectables via injections. So since these flouroquinolones, they have a good penetration into bones, they can be orally administered, and other drugs that would be parenterally administered, that would require to be used via injectables, that they can be replaced with these flouroquinolones having a good oral administration for the treatment of osteomyelitis caused by susceptible organisms. Since injectables results in patient discomfort, or they require sterile conditions, and they may act as potential risks to healthcare practitioners from bloodborne pathogens. They are, if there is another drug available which has a good oral administration, these injectables are avoided. So these flouroquinolones can replace other parenterally administered antibiotics for the treatment of osteomyelitis. They have a high potency and they have a long half-life. Adverse effects. GIT disorders, GIT disturbances like nausea, vomiting and diarrhea have been observed using these flouroquinolones. Then central nervous system disorders include mild headache, drowsiness, insomnia, dizziness and mood alteration. Anti-inflammatory drugs like ibuprofen may enhance the CNS stimulatory effects of flouroquinolones, and so these anti-inflammatory drugs should be avoided in presence of ciprofloxacin. They should not these flouroquinolones, they should not be used along with the flouroquinolones. Phototoxicity. The flouroquinolones like lomfloxacin, sitafloxacin and perfloxacin have been observed to cause phototoxicity. Arthropathy. Bone and joint arthropathies, bone and joint diseases have been seen occurring mainly in elderly patients when a steroid is co-administered. Hypoglycemia, low sugar level. These drugs, specifically gatifloxacin, can cause hypoglycemia or they may cause interference with diabetic drugs. Cardiotoxicity. Cardiotoxicity has found to be resulted in withdrawal of grapafloxacin and restriction on the use of sparfloxacin. Both these drugs were found to have a side effect of cardiotoxicity, and so their use has been restricted. But this cardiotoxicity may occur rarely with other flouroquinolones. This was all about our topic of flouroquinolones. Thank you very much.